What we're going to be talking about is a new technique that has been coming up in the last, it's growing slowly in the last decade and now just coming to play in the longevity field. And that is using mitochondrial transplant from an outside source, in this case, a bioreactor, to regenerate tissues. Okay. This was something that we started about 18 months ago, and we took on ourselves this idea that we'd start a project to look at this technique and see if it could be applied to longevity and to adult age-related diseases. And just recently announced that, yes, in fact, based on what we've seen so far, it is applicable and quite, in fact, extremely promising. I'll just mention that we have an amazing team. So we just brought on Dr. Tom Rando, who many of you know, as a science advisor. We have Dr. Michael Snyder from Stanford, who's head of the genetics department at Stanford. We have a group of principal scientists, all of whom are experts in their field and who've been doing this work for some time. So this is not something that's new. This is something that we're bringing together, this collation. So very quickly, the question we always start with is this: what if we could give a 90-year-old person the same mitochondria they had when they were 30? Okay. And of course, the real answer to that question is, when we ask any scientist, and they'll say, we don't know, because it's never been done. It's not a thing that happens in nature, obviously. But certainly, based on everything we're seeing, we see all kinds of opportunities for regeneration of tissue and really effects on longevity as well. And I'll be showing you some of the preliminary data that we've collected over this 18-month period. So what do we mean by mitochondrial transplantation? Well, really, in theory, it's as simple as that you go into a clinic, they take a bag of mitochondria. I mean, it's almost like a bag of blood, and they transfuse it into you. And of course, it's infinitely more complicated than that behind the scenes, but to the patient, it might seem that simple, okay? What happens is that because of the way that this is set up, those mitochondria actually transfuse into the cells, and a good number of them become adopted by the cells and become part of the mitochondrial network, okay? And then the net effect of that is that you're getting younger, healthier mitochondria, and more specifically, younger, healthier mitochondrial DNA that are supplementing your current inventory, okay? So the goal here is to really go after most age-related diseases, because most age-related diseases, one way or another, have some root cause in mitochondrial decline. And there's different ways you can fix a disease. You can go in after the fact and add some sort of therapeutic to try to fix the symptoms. Or you could go in and say, well, we're going to remove the root cause, which is lack of cellular energy, okay? Or you could do both. And I'll be talking a lot about that later, why you would do both of those things. So very frequently, this is not what you read in the science books about mitochondria. The official vision of mitochondria is that, well, they're just organelles. They sit in the cell. When the cell needs a new one, it just makes it, okay? As it turns out, that's actually not, that's oversimplified. In the last 10 years, there's been an enormous amount of research in universities all over the world, and they're finding over and over and over again that mitochondria transfer between cells. They float around in the bloodstream and get transferred via vesicles. Stem cells transfer mitochondria. They go between the cells themselves. So mitochondrial transfer, as the phrase down here at the bottom, which is a very important phrase, mitochondrial transfer is an evolutionarily conserved and pervasive biological process. It's happening in your body right now, 1 million times a minute, all over the place, okay? And there are profound evolutionary reasons for that. The bottom line is it's much cheaper energetically for a cell to get a free mitochondria than it is to make it internally. Making mitochondria is actually quite energy consuming because there's a lot of protein folding and all kinds of stuff that goes on. And so from a survival standpoint, if you're a little fish swimming around in the ocean 200 million years ago, and you could get free mitochondria that you're recycling within your own body, you're going to survive and you're going to prosper better than that other fish that doesn't have that adaptation. So all of this is fundamental to us as organisms. Okay, and of course, just so you know, all of this is our hypothesis of how the mitochondrial systems work. There's infinite amount of work done that needs to be done still to really elucidate all of the mechanisms here. And as I said, this all occurs naturally in the body. And here's a diagram that shows all the different ways that stem cells transfer mitochondria to and from other stem cells. And more importantly, stem cells donate mitochondria to ailing tissues. That's one of the things they do when they're regenerating tissues. That's one of the effects that stem cells have. So the issue with mitochondrial transplant, the idea is that we take mitochondria from an outside source and we transfuse them into your body, and that helps regenerate tissue. Well, if you're talking about doing that with, let's say, children's mitochondrial diseases, that's easy enough to envision how to do that, because if you have a child, it's very easy to get adults to donate tissue. And think of this like an organ transplant, okay? Organ transplants are very limited because it's hard to get organs. Well, mitochondria are limited. Nature does not give them away for free, okay? They're very valuable. So we don't have ready sources. With children, we can get them from adults, and that's perfectly acceptable. But if you're curing adult disease, you run into this problem. You're not going to go around and drain teenagers of their mitochondria, okay? That would not be ethically acceptable to anyone, much less the teenagers. So what our approach has been is that we have to figure out a way to grow these externally. And that's what our process is. We take stem cells from someone, or we take a line of stem cells. We grow them in a bioreactor, expand them dramatically. We remove the mitochondria from the stem cells. We coat them with a specialized artificial vesicle, a protein coating that has receptors built into it to target that mitochondria to specific tissues. That's a very important piece of it. And then so that we can effectively supply clinicians with a whole toolkit of mitochondria targeted at different organs. And then you send those to hospitals, to clinics, and that would be something that doctors would use regeneratively. And of course, that's very similar in many ways to stem cells. In fact, it's compatible with stem cells. Here's a little bit more detail about how our bioreactor process works. Very important not to trigger immune system, obviously. So what we've done so far is we've been working on 5 areas: skin, sepsis, CAR T therapy, neurodegenerative, and so forth. And the idea is to show that we have a comprehensive view of the whole body. Now, for example, we did work with regenerative brain tissue. What we know, our researcher that did this was up in Winnipeg. They know that energy production in the hippocampus of mice is something that is very commonly seen to go down. You see this difference between 1-month-old and 12-month-old. There's significant declines, okay, in the energy production in mice. So we went in and we did a bunch of injections of mitochondria, transfusions of mitochondria into mice. These were 1-month-old mitochondria that we injected into 12-month-old mice. So we're transfusing heterochronically. You know, it's the difference in age. Then we took out the hippocampus and looked, and we saw, lo and behold, that we had increased the mitochondrial expression in the hippocampus, which you don't see in any of the other cases. So this was a very unique result. This was a year ago. By the way, we're not the only ones doing this. Lots of people are doing this work in many labs. Then we went in at Stanford and we had the team there. It's the genetic retinal genetic ophthalmology department. We had them inject mitochondria into the eye of mice. And in this case, we have mitochondria going into this blue tissue, which is the retina. It's a cross-section of the retina. This layer here is the RPE layer. These red fluorescent things are the transferred mitochondria. We have a line of fluorescent mitochondrial mice. And we use those to track the distribution within tissues. So what we did is we did this injection. Lo and behold, the whole retinal layer, RPE layer, filled with these mitochondria. They transfer through the cells. They're shared between these cells. But not only that, then they start working their way up into the photoreceptors. And we have a lot of other photographs that they work their way up in the photoreceptors. They also work their way down into the optic nerve. Okay, so every time we inject them, they're moving out. We have a series of in vitro tests. We have an assay line where we're testing all of these concepts. And what we see here, for example, right here on the right-hand side, the dark blue is a control mouse. That's the energy levels, not a control mouse. These are cell lines. The control cells are at about a level of 4. This is all being done with the seahorse analyzer. When we inject mitochondria into them by incubation, co-incubation, we're doubling or tripling the respiration within the cell. That might be too much, by the way. If we did this as a time series, you'd see that this would begin to go back down again. The cell is, if it has too many mitochondria, it actually slows down and it lets them decline. So the mitochondrial system in the cell is extremely well controlled, as you would expect after 1 billion years of evolution. Cells are watching their mitochondria constantly, managing every aspect of them. Another interesting thing that we've done is one of the places that we found that you can get mitochondria and transplant them is by extracting them from platelets. Turns out that every time a platelet is activated or when it ages out at the end of its lifespan, right before it's taken to be destroyed or before it turns into a clot, it spits out its 5 mitochondria in these little lifeboats. It's like the Starship Enterprise popping out lifeboats and saving the crew. So what happens is that the platelets eject these vesicles, and the vesicles float around in the bloodstream. Well, why would the body do this? Okay. Well, we did a whole bunch of studies where we looked at these we took fluorescent vesicles that we extracted from the blood, from the platelets of one fluorescent mouse. We injected them a non-fluorescent mouse, and we looked to see what happened to them. What we found was within 2 minutes, most of those platelet-derived vesicles, lo and behold, had been sucked back up into all the other platelets and white blood cells and bone marrow of the local system. So fundamentally, it's a recycling mechanism. The immune system is constantly recycling its mitochondria. Every time it has one available, it's going to recycle it back into itself. There's an evolutionary process for preserving and conserving mitochondrial energy. And that may be why our immune systems work as well as they do. Okay. So fascinating, all kinds of fascinating work. And a lot of it we can't even talk about yet. We did just a flat out age reversal attempt where we took mice and we just kind of threw the kitchen sink at them. We gave them mitelet injections, vesicle injections. We gave them liver isolated mitochondria. We gave them young plasma. Plasma has tons of mitochondria in it, by the way, which you can find if you look for it. And what we found, we gave them lots of these injections, and these were older mice. We found that they just started acting young again. They literally, within an hour, they would be noticeably active. We saw that their fur would grow back faster. They had improved performance on a rotarod. And obviously, we'd like to redo this and do a lot more tests. There's all kinds of stuff we want to look at for this. And the limitation here is time available to do all this work. So this is a graph that shows all the different tests that we've done in various labs to look at the big picture of if this works across the body. And the bottom line is that it works. Everywhere we've looked, it works. Mitochondria are fundamental to the entire body and they go everywhere. So based on that, we're doing a series of products to boost the immune system, to boost the retina, and to regenerate the skin. This is still in very early stage, but we have orphan and fast track indications chosen for that. So let me just talk real quickly. I'm running out of time. I just want to say one quick thing. If you look at the difference between mouse aging and human aging, turns out that mice, when they die at 3 years old, we basically don't believe that they're really mitochondrially limited at that point. We think they're dying of other causes, and we're not the only ones to say this. And this really presents problems for the industry, because if you're trying to transition from mice to humans, what you find out is things that work in the mice don't work in the humans. And that's because humans are mitochondrially limited. We're one of the most aggressive preservers of mitochondria in the animal kingdom. That's the way I think of it. And so you're running into this completely different paradigm. So, for example, let's say we reprogram the nuclear genome, but we don't fix the mitochondria in the cell. Well, okay, so then we're going to have this really nice, beautiful mitochondria, nuclear genome, but we're not going to have any additional energy. And we know that mitochondria in an 80-year-old person are probably 70% degraded, which means you've got this really young nucleus. It doesn't have any energy to do the work that it now wants to do now that it's been reprogrammed, okay? Same thing with senescence. If you clear senescent cells and the mitochondria have very little energy, how are the mitochondria going to replicate new cells to fill that space that you just created? Okay. So, and in fact, we could even say if there were enough mitochondria, it very well may be that those senescent cells wouldn't have been created to begin with. Okay. Because senescence is frequently a lack of energy. So what we're concerned about is if we do all these various treatments to improve all these pieces, if we haven't fixed the mitochondria, we're going to end up with people who have wonderful, clean nuclear genomes, but no energy to do anything. They won't be able to get out of their wheelchairs. And by the way, there are diseases of teenagers, really awful diseases that are just like this. Everything about them is perfect except their mitochondria. And we know what the result of that is. It's not a good quality of life. So our argument is that mitochondrial regeneration is critical. We also know that doing it inside the body in place is very difficult because you're trying to fix something while it's working. That's hard to do, okay? It's thermodynamically very questionable if that's going to work. So our approach instead is, as I already said, let's do it in the outside bioreactor. We have an arbitrary amount of energy to input, and then we can get those mitochondria nice and clean and FDA approved, and then we inject them into your body. And that's a safer approach, okay? Let me just finally say that there's a lot of people, this is a very nice graph, diagram that we got from Quadrascope, and we're on here. We think that there's a lot of opportunities for mitochondrial transplant to support other vendors. We think mitochondria are a partner to these other things. And so that's the way that we look at it. And we welcome people who are interested in helping us get you past that translation wall from mouse to human. Okay. So that's it. And anybody who's interested in more information, also we have a place to sign up on our website for newsletters. Thank you very much.
All right, Tom. Well, thanks a lot. That was indeed fascinating. There's a lot that opened my eyes here to actual mitochondrial transfer as it occurs naturally that I wasn't actually aware of to the extent of it. So a couple of questions here. When really fundamental questions, when the mitochondria are released in a lot of these cells, is this primarily happening in stressed cells?
There is absolutely a difference in the receptiveness of cells based on whether they're stressed or not. If you have really healthy cells that don't need mitochondria, they frequently don't take them up. I'm sure there's a trigger somewhere that tells the cell whether it wants them or not, right? So yes, they are easier to transplant when you're stressed, but basically if you have someone who's 80 years old, all their cells are stressed at that point. So.
Mm-hmm. And related to that, so if cells are stressed, so in the body, if you have a stressed or damaged cell, is that a trigger for the mitochondria to be released from the cell?
Yes, I believe that's part of I think that's part of the process. Is that, for instance, if your muscle is decided it's going to atrophy and release a bunch of its nutrients to the system, which does happen with unused muscle, I'm sure that mitochondria are released in vesicles. And that's so they can be recycled in other more useful places. Okay, so
And related to that, those expelled mitochondria, do you know what the proportion is that are actually, quality mitochondria in the vesicles versus ones that are damaged as they are released?
Yeah, no, good point. I don't think there's much damage. Truthfully, as long as the process happens naturally, this is a very well-evolved system. It's extremely well-evolved. So I think it's pretty well done by the body. But I think that the ratio of damaged or mutated mitochondria to good mitochondria in the blood, according to all the studies, is representative of the body as a whole. Although organs are very different in terms of how many mitochondria they have that are healthy, okay? The body is allocating healthy mitochondria to the mission critical parts of, the heart has a very high ratio of healthy mitochondria right up until the end, whereas other tissues don't, okay, because the heart's mission critical.
Yeah, so when you talking about this and mitochondria being released, immediately my mind went to evolutionary biology, and I know this is a huge stretch, but the first connection that I had was algae being released from coral during coral bleaching, which is a symbiotic system where the algae or the coral is stressed, and then you have a release of algae basically fleeing essentially their symbiotic relationship, which
right.
It's a bit of an evolutionary stretch there, definitely.
No, I think that's very accurate. I think that the mitochondria have their own agenda. Yeah. They're smarter than they look, and they've been working this out for years. When things get rough, they jump ship. They look for another host.
Right. So, question from the audience here. How would the quality of mitochondria in a bioreactor be assessed, especially given that the majority of mutations in average mitochondria in various cancers are due to errors in mitochondrial DNA replication?
Yeah, absolutely. That is in fact the key question. And I can't speak precisely to it because a lot of it is confidential, but yes, there are mechanisms for assessing quality. That's why we have this lab where we're doing these. Basically, we have an assay line, and we're continuously looking at their ability to restore, not just whether they have a respiration, but whether they improve respiration in a test line. And we have people who do nothing but that. Okay. But then, of course, the question is, how do you improve the mitochondria in the bioreactor? And that is very tricky, but that's what we're working on. So.
And a question we have here, and hopefully I'm not garbling it and understand it correctly. If there's a sequestration, I guess, among cells of more damaged mitochondria, meaning that I guess some cells that are about to die retain the poor ones while the better ones get the lifeboat and go someplace else.
Right. Yeah, I personally think that is true. Yeah. In fact, I look at some people have said they think senescent cells are perhaps actually an evolutionary adaptation, where the tissue, let's say it's a hunk of muscle tissue, and the tissue is actually making decisions about selectively sacrificing certain cells and stealing all their mitochondria and giving them to everybody else, and then letting that one go into stasis with glycolysis. So, but these are huge questions.
All right. Well, that was really fascinating and eye-opening. And thank you very much, Tom.